for coming to the final lecture in our three-part seminar series, The Individual in the Genomic Era. And before I get started introducing our next speaker, I wanted to take a second to thank the group of people I keep forgetting to thank, which is the graduate students in Ecology and Evolutionary Biology, GREBS, because it was through hundreds of person hours that we were actually able to put all this together. So anyone who's in GREBS, if you could stand up and let the audience give you a round of applause, that would be great. Okay, there's my thank you. So it's my pleasure to introduce Dr. Carlos Bustamante to you tonight. Carlos did his undergrad and got his master's and his PhD at Harvard University. After his PhD, he studied at Oxford University before joining the faculty at Cornell University. A couple years ago, he decided to leave the Ivy League and join the Pac-10, so he's now a professor at Stanford. And recently, he was awarded a Genius Grant from the MacArthur Foundation for the work that he's done and that he's going to present to you today. Dr. Bustamante studies the patterns in the genomes of individuals that are generated by mutations over evolutionary time. He's then able to use the patterns that he discovers to understand the evolutionary history of a species or many species, and also to trace how these mutations change the phenotypes of individuals. Today he's going to talk to you a little bit about this work that he's done in both dogs and humans.
Great.
Carlos?
Thank you very much. It's a tremendous pleasure to be here today sharing with you some of the work that we and others have done in trying to understand the genetic basis of complex traits. And as a way of motivating this work, if we think about the revolutions that have happened in the last 10 years since the sequencing of the Human Genome Project, it's really breathtaking to think about how genomics is now basically being applied to every aspect of your life. And if we think about the next 10 years, I'll argue that, in fact, it will continue to permeate almost every aspect of how we think about different human populations, how we think about food production, how we think about control of pests, how we think about our pets. I mean, it really is something that has now opened a huge floodgate and has the promise to really aid humanity in lots of different areas. For example, it can begin to disentangle the genetic basis and contributors to many chronic diseases. So, for example, if we want to understand the genetic basis of diabetes or cancer, heart disease, epilepsy, these will eventually become problems where genetics tied to careful phenotyping and life history information is going to really promises to make an important contribution. Likewise, it's already kind of the standard by which we think about infectious diseases and tracking infectious diseases. It ends up being a problem of understanding the genetic basis of the pests and how they're moving through populations. Likewise, in agriculture, where we've worked in rice and cattle, it is really being spearheaded at the forefront of developing tools so that we use the natural genetic variation that exists within species. So really moving away from genetically modified organisms where you're introducing genes that come from different species that may be quite different, and rather harnessing the genetic variation that exists within corn or rice or cattle to really drive the phenotype in the direction that one wants. And what's really key to thinking about these problems is that they're essentially marriages of Darwinian evolutionary theory to Mendelian populations. That is, how do we think about the problem of inheritance at a population level? And the implications are huge, right? So the goal, why we as a society are investing a huge amount in this area, number one is because we think it'll benefit. And there have now been arguments about whether the investment in the Human Genome Project was worthwhile. And there are these really pernicious articles that are being written that I think are doing science a tremendous disservice because they fail to really quantify all the externalities that have happened from the genomics revolution. And when that's done, economists have argued that in fact the investment has been 10 or 100-fold returned in terms of what was invested to figure out the first human genome. And I'll give you some examples of that today. And as I said, there are implications both in agronomics and in medical genetics. So in particular, I want to think a little bit about why is evolution important to this? Why do we want to think about evolutionary processes problems? And the answer is because they are fundamentally problems of how populations are changing through time. And some of you may have seen me fiddling with my slides earlier. So if we think about today how much it costs to think about genetic variation, the investment in the Human Genome Project has dropped this precipitously. So to score about 500,000 places in the human genome for DNA sequence variation now costs under $200 a sample. And I had come up here, and this slide said about $10,000 to $50,000 because it was from the last time I had given part of this lecture. And just to update you, this is an email from today. Actually, it's today's date. And this is from one of the companies that now does human genome sequencing routinely. And they sent me this email. This is actually forwarded to my lab's email list. And they're saying that if you have between 0 and 50 human genomes you want to sequence, they'll do it for $5,000 a sample. Up to 250, they'll do it for $4,000 a sample. Between 500 and 1,000 human genomes, it's $3,500 a sample. And let's talk if you have more than 1,000 human genomes that you want to sequence. Okay? So really, it's just dramatic to think that basically all of you in this room will likely have the opportunity to get your genome sequenced for next to nothing. And how are you going to make use of that data? How are people going to make use of that data not only of human genetic variation, but of other species? And in my group alone, we've applied it to cattle, rice, grape, and I'll tell you mostly about the work done in dogs, but if we think about it, the reason we can do this is because fundamentally the problems are very similar. We want to quantify why different groups are different from one another, how different are they, and we'd like to use this in association with information about how the groups vary phenotypically to identify genes underlying diseases, say in humans, or milk yield in cattle, or seed size in rice, and so on. Okay, so the structure of today's talk will begin by talking about a project called the CanMap Project, and the goal of the CanMap Project is to use genomics technology to understand the genetic basis of the phenotypic variation in the domestic dog, and then hopefully we'll have time to tell you a little bit about some of our latest work in applying the same theories and approaches to identifying the genetic basis of blonde hair in an isolated human population from Melanesian. Okay, so I want to begin by telling you about this rare animal. I understand these are very rare here in Oregon in particular, right here in Eugene, the domestic dog. As many of you know, dogs are descended from the gray wolves, and in the last 10 to 15,000 years have dramatically changed in shape, size, and conformation, and they now range from dancing Irish wolfhounds to Park Avenue princesses to chihuahuas or St. Bernards that'll bring you a little bit of hooch if you get lost in the Alps. And for a long time when I gave this talk, actually, I didn't realize this here is not a real dog. That's actually a little statue of a dog. It gives you a sense of the phenotypic variation. In fact, dogs are the most phenotypically diverse mammalian species. They differ by 2 orders of magnitude. It's pretty astounding if you think about the size range. It would be equivalent to having humans that were about 2 feet tall or 20 feet tall. Okay, so the project is a project that we began several years ago called the CanMap Project. It involves Elaine Ostrander at the National Human Genome Research Institute, Bob Wayne at UCLA, and then later John Novembre and Adam Boyko, who's a postdoc in my lab, who's actually going to go back to Cornell. So the goal of this project was to take and apply genomics technology, a very standard approach for looking at genetic variation in dogs, and systematically examine 900 dogs from 83 different breeds, 200 wolves. We've actually now expanded this to several hundred more wolves, initially 500 African village dogs, 50 African village dogs, which we've now expanded to about 2,000 village dogs from throughout the world. And we've genotyped all these dogs on the same technology. That is, we have the same set of genetic markers in these dogs that we're able to quantify and compare, and then looking at patterns of variation that are fixed among dogs that share a trait, begin to map the genetic basis. And I'll walk you through how that's done. But schematically, if we want to think about what's the big picture, the reason this project was motivated, what excited us about it, is that dogs come in very well-defined breeds. Okay? So the variation in phenotype within a breed is often very, very small because of the tremendous sort of breed standard. So furthermore, many breeds share a trait that is often of interest. So for example, we might think about achondroplasia, or foreshortened limbs, or dwarfism. And we might imagine that these dogs, the sort of dachshund and corgis and basset hounds, will all have legs that are much shorter than one would expect if they were a normal-sized dog, may share a common mutation. So if we look for regions of the genome of these dogs that are similar and different from other dogs, then that might give us a genetic clue as to why they have achondroplasia. Likewise, we might want to focus in on brachycephaly, or the foreshortened faces of bulldogs or dogs of that variety, griffins and so on, and compare it to, say, dogs that are domesticated, that have long snouts and sighting herds and so on. And again, all those dogs will only be similar in the region where they share the mutations that underlie the same phenotype. And in particular, one of the traits I'm going to be telling you about today is body size variation, and how we've begun to piece together the story of dog body size variation. So in order to do so, I just want to walk you through some of the basic principles that we're going to use. One of them comes from quantitative genetics, and it's known as association mapping. I'll give you a schematic for that. I just want you to sort of hold those two ideas in your head. One is association mapping, and the other is selective sweep mapping. And the idea behind association mapping is actually quite intuitive. So let's imagine that we want to compare two populations. And let's imagine, I use this example in talking about medical diseases as well. Let's imagine you want to compare cases, so individuals that have a trait versus controls, individuals that don't have the trait. And let's imagine that we're thinking about the whole genome, and each of these lines essentially represents a place in the human genome, or in the dog genome, or whatever genome you're studying, where you have two potential forms of the gene, so-called alleles. And we often represent these as big A or little a, and the name of the game is simply to compare the frequency in cases versus the frequency in controls. It's the same thing that happens when we think about elections, right? We want to compare how many people voted for the Democrat versus the Republican, and that's going to vary from county to county. Okay? So you might imagine in this place in the genome, cases have about 50-50 frequency of allele, big A versus little a, and controls have about the same. So there's nothing really exciting going on. So now we simply move on to the next county, or the next part of the genome. Now, it's going to vary, but here it's about 80% to 20% in cases versus 83 to 17% in controls. Again, there's nothing really exciting going on. Cases and controls are about the same. Okay? So we're going to play this game until we hit upon a region such as this, where the frequency of the big A allele is 70% in cases, but only 40% in controls. This is the very basic idea behind genetic mapping. You simply scour a bunch of genetic markers and compare the frequencies in the two groups, and there's some statistics behind this, but at the end of the day, we might be able to say that if you carry this mutation, you're about 3.5 times more likely to be a case than a control. That's the notion of association mapping. Okay? Now, because we're talking about organisms that have been under natural selection, or in this case, artificial selection, we might also bring in a very key idea from evolutionary biology known as selection mapping. So if we think about selection mapping, the key idea behind selection mapping is if a mutation has changed quickly over time because of the frequency being driven by selection, then that's going to leave a characteristic signature in the genome. Again, I'll walk you through the example in this case. So if a mutation is subject to strong selection, it's going to fix quickly in the population. That means that you're going to have a reduction in genetic variation right around that target of selection. And because domestication and breed formation were such strong events, you're talking about some of the strongest selection that populations have been subject to, this likely happened very quickly over short time scales. So the hypothesis is they might be hitting the same genetic variation. So let me walk you through this example. Let's imagine each of these is now a chromosome that one wants to compare. These are genetic markers, and so we have what's known as variation, right? Some individuals carry an X, and some don't carry an X at this position. Again, akin to our big A or little a. Then there's a beneficial mutation that arises. These are known as haplotypes, so the two different colors. We've got red haplotypes and purple haplotypes. Here we have a beneficial mutation. Here's another mutation that's linked. So we have link site here, a link site here, and then we have this mutation that arises. So the idea behind selection, of course, is that this mutation, after a couple generations, is going to increase in frequency. Does that make sense? Well, what happens? You drive out the other mutations that exist in the population. same idea that happens whenever you select for insecticide resistance or drug resistance. The individuals that carry the beneficial or selected mutation, so if we're thinking about those dwarf dogs, the dachshunds that carry the foreshortened limbs, they're going to breed more of those until the other ones are going to be less of them in the population. So if this happens really, really quickly, well, you don't have to be a professionally trained evolutionary biologist to figure out that everybody around that region is just going to have that purple haplotype, right? I mean, it just kind of stands to reason. And if we walk out from that region of the genome, though, there may be some recombination that then recovers variation. So those are the two ideas. Number one, we're going to compare populations that differ in the trait, so cases and controls. And number two, we're going to look for regions of the genome where there's a reduction in variation in one group versus the other. So we've now successfully applied that idea time and again in dogs to identify genes underlying morphological traits. And this is one of the first, one we call dramatic examples of this. This is the paper we had in Science several years ago that identified a mutation in a gene known as insulin growth factor 1 that is carried by all tiny dogs. Every tiny dog breed that we looked at carried this mutation. And I'll walk you through the logic of that paper and how it relates to the broader CanMap project and how we've now started to look for this mutation in all the world's dog populations. So this story begins, like all good stories, about a family. And this particular family was actually Portuguese water dogs. So Portuguese water dogs, when I began to tell the story, were actually quite nobody had ever heard of Portuguese water dogs. Now everyone knows about Portuguese water dogs, because who's got a Portuguese water dog? Obama. It's Obama's dog. And it turns out, I don't know if Obama chose it for this reason, but they come in all kinds of shapes. And this is what one might call Photoshop-induced body size variation. Big amount of variation in these dogs. And the interesting thing about Portuguese water dogs is that there aren't many of them. So you can draw this big pedigree that has all the Portuguese water dogs in it. And these guys from Utah, Gordon Lark and Kevin Chase, have been doing this for years. They've been looking at these really carefully detailed pedigrees with genetic markers. And when you do that, and you look at body size variation on the pedigree, you find this really strong association, and that's what this is supposed to represent. So this minus 10 log p-value is just a statistical way of giving evidence for the association. So the smaller this p-value, that is, the more the minus 10 log p goes in this direction, the stronger the association. For this part of the genome, in fact, it's chromosome 15 in dogs, to be associated. Now, the important thing about this association is that because it was just one big extended family, the region that was associated was huge. It was megabases. What that means is there were hundreds of genes in there. And you couldn't really pinpoint a single place that was associated. So this is where this kind of collaborative team came together and said, well, why don't we take a look at that part of the genome across a bunch of different dog breeds? Let's take, say, tiny dog breeds, giant dog breeds, medium-sized dog breeds, and just sequence them. Let's look at a couple of genes in that region and see what we find. So we began by essentially doing exactly what we do with association mapping. Instead of having cases and controls, we have big versus small dogs. You look at how many carry the big A, how many carry the little a, and you do your statistical test. And that's what we ended up doing. And when you do that, you find this really striking association. So again, here's our minus 10 log p-value with this set of markers here. And what was amazing is, instead of having this association go on for megabases over hundreds of genes, we could nail it to a single region in a single gene. We basically got it to within the two parts of a gene called insulin growth factor 1. So you might ask, well, what do we know about insulin growth factor 1? Turns out to be an awesome gene of mammalian biology. You knock out insulin growth factor 1 in mice, you get mice that are 40% as big. There are humans that walk around with mutations in insulin growth factor 1, and they're about 60% as big. Right? So it makes a ton of sense. So this is a gene that's involved in mouse body size variation, it's involved in human size variation, and now we've linked it to dog body size variation. The other reason this was really cool is because it was one of the first demonstrations of applying this notion of the selective sweep. So FST is a measure of genetic differentiation. It's a way we quantify how different groups are. So when we this immediately suggests a hypothesis that what happened was that we had a mutation that arose that on a background that then gave rise to small dog breeds. Okay? We can now test this. And the way we test this is by comparing the frequency of that mutation now across 143 breeds. So we've started with one breed. It gives us a place in the genome that looks interesting. We then look at, say, 20 dog breeds to map it down to a reasonable region. We then find one place in the genome that looks to be really strongly associated, and we can now go and test it in 3,000 dogs from 143 dog breeds. And what we're essentially trying to do is ask, does the frequency of this mutation correlate with the average size of dogs in that breed? So each dot here represents a dog breed, and here's the frequency of the mutation. And you don't get data like this very often. I mean, the association here is actually pretty strong. All of the small dog breeds, so those that have male body sizes between, say, 4 and 16 kilograms, they're all basically fixed for this new mutation, whereas all the dog breeds that are above 25 kilograms, basically none of those dog breeds have the mutation. It's really strikingly strong evidence. The other thing we can do is now go back and look at wolves. So what allele do wolves have? It turns out wolves only have the big dog allele, which makes sense, right? It's consistent with the mutation having arisen, giving rise to small dogs, and so it's very rare in wolves. Furthermore, what really kind of cemented the deal is that when you look at the small Portuguese water dogs, they actually carry exactly this same mutation, this same looking chromosome, whereas the big Portuguese water dog carry the same, they basically look like Irish wolfhounds at that part of the genome. So it's a really kind of, bringing together lots of different pieces of evidence. One is this percolation of the allele in the Portuguese water dog pedigrees. Two is the variation, the sequence variation across a limited number of dogs sequenced from different dog breeds. And the third is this replication across lots and lots and lots of dogs of this particular mutation. So this was great. We were really proud of ourselves. This was, we were patting ourselves on the back for a long time. The only problem with this is that it took like 3 or 4 years, millions of dollars, a big team of people, and at the end, we got one mutation. Right? It's not a very efficient way of going out and trying to knock out all the different mutations that are involved. But if we think about humans, for example, in humans, we know of 180 places in the human genome that are all associated with height. They explain only about 10% of the variation in height, and we still don't know how to trace down those mutations. So no one's going to give us money to start tracing this down in dogs in any systematic way like that. Right? So we had to come up with a really kind of rapid-fire way of doing it. This is where genomics came in. So in genomics, we can take, and this is an example of one of these gene chips, we can now use what essentially looks like a microchip, and on that microchip we can use the patterns of genetic variation to simultaneously look at hundreds of thousands and now millions of places of the dog genome and compare them across all the dogs. The other thing we can do is do this for lots of different traits at the same time. We take 1,000 dogs, they've been measured for a bunch of different traits, or they belong to breeds where we know the traits in the breed, and then we can look at dozens and dozens of traits at the same time across the same genetic data. So when we do this, the first thing we see here is, again, that same analysis for insulin growth factor 1, but now we're looking at all the data from, in this particular case, 60,000 places in the dog genome. The first, which is great, we replicate insulin growth factor 1. That's one of the good moments for us. All right, we got that one. That wasn't a false lead. But then we see that there are a couple of other places that look really promising. And in this case, we're using one algorithm. In the second case, we're using a second algorithm. But they all seem to kind of line up in terms of where the associations sit. We can then begin to really piece together this picture. So here is that same figure, but now overlaid genes on top of them. So there's a gene called SDC2, which it's in this region. SMAD2 is in this region. Well, both of those genes you find in cancers. Whenever you're comparing a cancer mutation to normal, it turns out they're often genes in these mutations in these genes that are leading to cell proliferation. So that makes a lot of sense. HMGA2, well, that's a beautiful association. Why? Because it's the #1 associated gene with height in humans. So here you're taking what we're learning from humans, We're kind of verifying it with dogs as a model system, and you're now setting up this feedback loop, right, where what we're learning from dogs could help us then someday annotate the human genome as well, our insulin growth factor mutation and so on. And we can begin to piece these together, and now I don't expect you to be able to read this in any way, but each of these rows is now a different trait. So instead of just looking, say, at average body size, we can look at leg width, we can look at head length, we can look at a bunch of different traits. Here are a bunch of other regions that are associated. All I want you to take away from this is that oftentimes, many of these mutations, they're impacting not just one trait, they're not just impacting body size, but of course because body size relates to leg length, and it relates to shoulder height, and it relates to back length, and so on. Many different traits are kind of scaling with these same mutations. So we can begin to piece apart and ask, well, what kind of mutations are only impacting the height and not, say, the weight and that sort of thing. The other thing we can begin to do is build multi-gene models. That's one of the things that's been particularly hard in humans. So I just mentioned when we try to look for genes that are associated in humans, we find that we can only really explain about 10% of the variation, which isn't very good. If we look at the mutations of insulin growth factor 1, R squared is a measure of how much of the variation you're explaining. So just with insulin growth factor alone, we're explaining about 32% of the variation, which is pretty good. Again, each dot here represents a breed. So here are all the breeds that are fixed for one mutation, here are all the breeds that are fixed for the other, and here are the segregated breeds. The amazing thing is we can now take those 4 or 5 regions that are associated, and now, if we put those together, we're explaining about 86% of the variation, which is pretty dramatic. That means that with 4 to 5 regions of the dog genome, I can put them together and I can predict the size of your dog. Okay? There are very few organisms on Earth for which we can do this. In fact, I think dogs are probably the only one. Right? And the reason is because of selection. Because selection has just been so dramatically strong, and because it's been so dramatically recent, it's basically hitting these same mutations, and you're just stitching them together to make different dog breeds. Okay? You can almost imagine an app, right, that you can put on a website to order a dog, slide up the mutations, and bam, comes out the dog that you'd be able to dial up by looking at these different mutations. In fact, we're working with National Geographic exactly on an app like that, so you can look through yourself these data and figure out how they could be leading to different combinations. Okay, so that's kind of cool, that's kind of interesting. But one of the big criticisms from evolutionary biologists is, well, so what? We know dogs have been under selection. There may be not a real model system. How relevant is this to what's maybe going out in nature? Well, what's really cool about dogs is that aside from being our pets and companions, they're also eking out an existence on the edge of human settlements. That means that dogs that aren't being directly taken care of by humans are under a tremendous amount of selection. Right? They have to adapt to be able to live near humans. They were, in some sense, the first domesticated species. They were following humans as we were settling over the last 100,000 years, all of the continents. So they've had to rapidly keep up with us. And one of the things you might ask is, well, do these mutations that you find in the breed dogs, do they predict at all dog height in any place in the world? And so this is a project that was started several years ago by Adam, who was a research associate in my lab. And Adam came to me with a really wild idea. He said, Carlos, why don't we just go to, this is when we were doing the IGF-1 thing. He said, Carlos, why don't my brother and I go to Puerto Rico and get us some dogs? I said, are you serious? He goes, yeah, we'd sample some dogs and we'll see whether the little dogs carry IGF-1 or not. And so they bought some dog catching equipment and some gloves, and they went out and they came back and they sampled like 12 dogs. And I said, okay, well, that's good enough for me. So Adam then got his brother and sister-in-law, and they had just gotten married, and they were anthropologists. And like good anthropologists, they just want to spend their time out in the field. And like all anthropologists, they never have any money. And so they came to me, and I said, well, I think this could be mutually beneficial. I'll pay for half your honeymoon, you get me some dogs. And so they went on their honeymoon through Africa collecting dogs for us, and what came out was a really kind of cool way of beginning to compare the maps that we were making from breed dogs to village dogs. So then they wanted to go on a second honeymoon, and then we got dogs from home. Okay, so let me give you some of what this village dog data begins to look like. Every dot here is a dog. Males are based on our genetic model. And again, the correlation's pretty good. I mean, that does a good job. This is for breed dogs. Now, this is the same analysis, but for village dogs. So these are dogs, they're all eating Purina, they're all living in Eugene, Oregon, or wherever, eking out a nice existence. And then these are scrappy village dogs, eating God knows what, you know, of different ages, at different times, yet we can still predict pretty well. I mean, it's not as well as in breed dogs. We can still predict pretty well how big those dogs are going to be based on 4 places in the genome that we identified from breed dogs. And that really means that many of these village dogs, these are mutations that are pretty old. Okay? For them to be in breed dogs and in village dogs all over the world, we've also analyzed these village dogs to ask, are village dogs just a mixture of breeds? And they're not. I mean, some are, obviously, but the vast majority are totally different. They have totally different looking genetics. And so what you're really looking at is human selection over 10,000 years, hitting the same few mutations that were pretty common at the time of dog domestication. The other thing we can ask is, well, how well does it predict in different parts of the world? And this is where the story got really interesting. It's a little bit more complicated, but follow me through it. So if we look at the ancestral homozygote, so these are dogs that carry the wolf allele. These are dogs that carry the new derived mutation and are from India, say. Well, the relationship there is really nice. So the ones that carry the ancestral allele look like wolf size, so on average they're about 58 pounds. But if they're derived, they're about 31 pounds. That's a pretty damn nice correlation. Now we look at the same thing in the Middle East or Egypt, same relationship. We look at dogs from the Americas and Europe, and this is particularly strong, but of course that makes sense. These are the dogs that are particularly likely to be mixtures of breed dogs. Now, in Oceania, not so good. Vietnam and Borneo, not so good. Africa, okay. And then within Labrador Retrievers, it doesn't really hold. But of course, you might expect Labrador Retrievers to basically have only one form of the gene, so it's not what's the reason for Labrador Retrievers. So what gives? Why might this look so funny? Well, the other thing that's kind of odd is if we look at the dogs that are the smallest, they're the Oceanian dogs. So the smallest dogs actually seem to have the littlest effect of this gene. So that's just showing the same relationship. So let's just look now at the frequency. Well, here's the ancestral allele. Here's that wolf allele. All the European dogs basically carry the derived allele, which makes sense. And you're looking at that frequency. So it's varying all over the place. In the Americas, all these dogs actually are derived from European dogs. We've done some really thorough collecting in Native American communities, trying to find native dogs. We've yet to do it. We're now starting to look at ancient DNA to see if we can find some remnants of ancient dogs. But I'll draw your attention out here. So here's these Oceania dogs, the smallest dogs in the world. We've got some from Papua New Guinea, and they just don't look like they carry a big frequency of the derived allele. Why could that be? Well, if we think about dogs and dog domestications, there have been 2 major hypotheses. Dogs were domesticated in China. No, dogs were domesticated in the Middle East. No, dogs were domesticated in China. No, dogs were domesticated in the Middle East. Right? So there's been this debate in the community about where dogs were domesticated. Folks, dogs were probably domesticated more than once. Okay? They're probably domesticated in China and in the Middle East and maybe down here and maybe over. All right? As a friend of mine says who studies coyotes, they should all be called canis non-discriminatus, right? Because they're very happy to mate among themselves, right? So what we're probably seeing are these signatures of differences in domestication that are telling us about the number of different times dog breeds were domesticated. The coolest part of this whole story is that when you look at Shih Tzus, in fact, all Shih Tzus carry the derived allele, and they're the ones that have the biggest signature of selection, which is consistent with Shih Tzus having come from a Chinese origin and then subsequently made smaller by breeding with European breed dogs to bring in that insulin growth factor 1 mutation. Okay, we can now take this same idea and invert it on its head. So we began by saying, let me take all those traits I'm interested in and compare them across dog breeds. Let me look for where the small dogs are different from the big dogs, where the dogs with little legs are different from the dogs with tall legs, and so on. We can also simply ask, almost in a kind of unbiased way, where function of allele frequency. So these are the things that are both really common in dogs and really different among dog breeds. And so one of the most differentiated regions, should be no surprise to you now because I've been telling you about it for half an hour, insulin growth factor 1. And here are all the breeds that are fixed with the derived haplotype. Here are all the breeds that are fixed with the ancestral haplotype. So it's a nice kind of positive, true positive that we can use to root our thinking. Now we move on to the next idea, which is that dogs aren't showing this really strong breed group relationship, but rather this kind of adaptive radiation. So if we take different dogs from different dog breeds and try to build a big tree of how they're related, we see that we can get all the dogs from a given breed to be each other's closest relative, but then the whole thing kind of collapses. So it is not the case that all the small dog breeds are more closely related and all the big dog breeds, but rather there's this kind of adaptive radiation of dogdom that allows us to do this. So when we now look at the next set of regions that are showing strong differentiation, bam, they hit on genes that are involved in hair and pigmentation. So we've mapped genes for coat color, for furnishing, for fur length, for coat color. And this is giving you basically that same idea. We can basically take 3 different genes and in different combinations predict whether the dog will have short hair, wire hair, long hair, curly hair, just the combinatorics of putting together those dog breeds, and we can basically predict everything from the Basset Hound phenotype to Bichon Frises to Irish Waterfang, right? With 3 different genes for coat color. So we'll play the game again. What else do we find? Well, this is one region that was the most differentiated region. Okay? This is a region all on chromosome 10, and here are the dog breeds that are associated. To help you out, here are the pictures of the dog breeds. What do you think this gene underlies? Ears. There you go. Prick ears versus flop ears. So all these dogs have stand-up ears. All these dogs have little floppy ears. Well, it turns out floppy ears are a key hallmark of domestication. Most mammals have prick ears. When, there are very few that have these, and the ones that do are all kind of domesticated animals, right? So it's kind of this hallmark of domestication that people have implicated and is thought to be involved with a general neoteny or trying to keep animals in a juvenile state. Okay, and it turns out that we've now mapped the gene for that, which is MSRB3, by looking across different village dogs. So if we put this whole story together to make a very long story short, if we look for those regions of the dog genome that are most differentiated, we basically find all the genes that define the dog breeds. We find genes for coat color, for body size, for foreshortened legs. We find for skull width, for foreshortened faces, ear floppiness, everything that basically you would imagine humans having selected. What's really fascinating is that these regions are totally different than what you find when you look at humans. So when you try to play the same game and you look at different human populations and you try to ask, what are the genes that seem to have been the most selected for in human populations? They're all involved either in skin pigmentation, and I'll tell you a story about that, in disease resistance, or in human metabolism. So genes involved in the ability to digest milk, genes involved in whether or not you're susceptible to malaria, or, and this is part of the paper that we had several years ago, genes that are involved in skin pigmentation. This is the signature of selection across one of the KIT ligand locus that we and others have implicated as having been particularly important in very recent human evolution. So if we think about this idea, and we think about what we're learning from man's best friend, it immediately raises the question of how can we turn this notion of looking for signatures of selection as a way to scan down and find genes that may be underlying really cool human traits. So it relates to this broader philosophical problem, and some of you may have seen this paper, as to whether what's happened in humans has been a region of hard sweep, that is what we saw in dogs, that whenever we're selecting on the same trait, we're basically getting the same set of genes, or whether it's been more of a polygenic adaptation. That is, selection has been trying to solve the problem, say, for skin pigmentation in different continents, and you're hitting upon different potential solutions. So in order to really think about that problem, we've started to collect human populations that segregate for really interesting and cool traits that may be subject to selection or may at least be kind of interesting to study. And so I want to end by telling you this sort of really interesting story for us about trying to understand the genetics of human biodiversity, and I'll focus in on these beautiful kids who live in the Solomon Islands, which are off the coast of New Guinea. And you look at these kids, and you see that they have this, some of them have this beautiful blonde hair. And you say, okay, well, where would that blonde hair potentially come from? So you might see the serviceman's jacket and go, okay, I've got a hypothesis about where that gene may be coming from, a kind of Captain Cook hypothesis about genes. And so you might imagine, for example, that the reason this boy has blonde hair is the same reason some of you in this room have blonde hair. So if we take a step back and just think about the great human diasporas, humans, as we know, we have our common origin in Africa, and through a series of migrations over the last 100,000 years, have really populated the rest of the world. And the incursions, particularly in Melanesia and Polynesia, some have happened quite early, so somewhere about 40,000 years ago, humans are already beginning to colonize this part of the world. So the alternative hypothesis may be that, in fact, it's a different mutation. But it's not the same mutation that gives rise to blonde hair. So we need to come up with a way to really tackle and assess that. So in order to do that, the only way you can do it is to go out to the field and actually collect the samples. The other reason we did this work is because we're really, really concerned by some of the tremendous biases that happen in medical genetics. So 95% of medical genetic studies have only included participants of Northern European descent. Let me repeat that. 95% of medical genetic studies have only included participants of Northern European descent. So for us, it's not only an issue of understanding interesting questions of human biology, it's actually a question of equity and how do we make sure that we don't just invest genomic resources in populations that are already medically well served. And so it's kind of a way to begin to say, you know what, when we go into these other populations, we can also identify genes that are important. This is the first study that had really been done for any trait in any population in this region. Okay, so we began by taking 50 kids that had blonde hair and 50 kids that had dark hair. And these are the postdocs from my group who led this project, a guy named Sean Miles, who went out and phenotyped these kids. And to be honest, when we had tried to explain this project to medical geneticists, they said, you guys are totally crazy, right? Whenever you go out and we study the genetic basis of any disease, we need 2,000 people with diabetes and 3,000 people without diabetes. 50 kids with blonde hair and 50 kids with dark hair, there's no way you're going to find anything. All right. Well, we were very happy to prove them wrong. This is our plot of association. These are often called Manhattan plots. In our case, we like to call it our Dubai plot because we have this really beautiful single peak of association. And what's even cooler is that this peak is only in one part of chromosome 9. It's this very small region of chromosome 9. And you can't read the label here. There's only a single gene. Turns out that gene is called TERP1. And TERP1 makes a tremendous amount of sense as a potential gene that may be important for this phenotype. Number one, it's involved in melanin synthesis. Well, that's good. Number two, it's involved in the maintenance of the melanosome structure. It affects the cells that produce pigmentation and how they proliferate and cell death. And then the real sort of other smoking piece of evidence is that there are people described with knockout mutations of this gene. And in fact, they have a very particular form of albinism. It's called oculocutaneous albinism type 3, or rufous albinism. And the reason it's called rufous albinism is because it occurs in individuals with dark pigmentation, and it leads to a red phenotype in the albino individuals. So it makes a tremendous amount of sense that this might be involved in the skin pigmentation that we're seeing in the Solomon Islands. So we went through. For those of you who aren't familiar with this, there's a way of looking for signatures of selection. Here is the signature for the allele that carries the mutation. Here's for the other allele. This is often taken as a very strong signal of selection. For anyone who's interested, I can tell you more about it. It's the equivalent of looking heterozygosity. Furthermore, we can go through and actually try to map this down to a functional mutation. So if we think about the gene, here's TRP1. It only has a handful of exons, so the parts of the gene that encode protein. So we sequence it in 12 cases and 12 controls. And again, this is kind of our really lucky project. It turns out that all of the people who were blonde had the exact same mutation. All of them. And in fact, they were homozygous for the same mutation. Both the copies they got from mom and dad had the exact same mutation. It was the only mutation in the regions that differentiated cases and controls. Furthermore, it's a mutation that impacts the amino acid. So it's a mutation that leads from an amino acid called arginine to an amino acid called cysteine. For those of you who are aficionados of biochemistry, know that whenever you substitute in a cysteine, you've got to be careful because cysteines make disulfide bonds. So one way to see whether this might be an important mutation is to look at other species. So when you do that, you see that so our kids have the cysteine mutation, everybody else has arginine, and when you look at rhesus has arginine. When you look at mouse has arginine, dogs have arginines, elephants have arginines, possums have arginines, chickens have arginines, and zebrafish have arginines. So again, you don't need to be a professionally trained evolutionary biologist to say that if every other vertebrate species you've looked at has arginine, this might be an important part of the molecule and maybe functionally important. We can then go through and replicate. Again, if we think about the way we did this in the previous example, you begin to hone down and then increase your sample size. In this case, we had 1,000 kids that we had looked at, and for each of those 1,000 kids, we could ask how many are arginine-arginine, how many are arginine-cysteine, and how many are cysteine-cysteine, and you can build a big statistical model. All I'll show you is here's the distribution of hair pigmentation. Here are all the blonde kids, so they're like 2 standard deviations away from the mean. It's about 10% of the population is blonde, and if you put in and build a model, you see that our prediction is that substituting the mutation basically jumps you from here to here. So it's a single real mutation that basically is knocking you from having this kind of normal dark hair to having the blonde hair. Furthermore, it seems to be a totally recessive trait. Whenever we saw it, we only saw that the kids were homozygous or the cysteine mutation. It's a really kind of trap shut tight kind of argument. Furthermore, when we characterize the frequency, it's at exactly 30% in the population. 10% of the population is blonde. So again, you just multiply 30% by 30%, and that gives you the frequency of blonde hair. It's really tight. The other thing that we can do is begin to look at different populations and characterize the frequency. We never saw this mutation in any other population except for some Fijians. When you look at the Fijians, they're blonde Fijians. So you can compare against the Chinese, the Yoruba, or the Seth. So these are individuals from Utah. These are individuals from Ibadan, Nigeria. These are individuals from Beijing, China. Here are cases and controls. So it essentially is unlikely to be a mutation that came in from Europeans. Rather, it's a mutation that predates and may well be about 10,000 years old from the dating that we've done. This is further evidenced by, let's look at other populations. So this is what one might call chemical blonde polymorphisms. Nonetheless, we know a lot about why blondes are blonde, at least in European populations, and they all carry mutations either in OCA2 or in the KIT-like. They don't carry mutations in TRP1. So it's a really kind of neat and solid story. To put the last bow tie on it, when you knock out the gene in dogs, it also has an impact on coat color. In fact, in Newfoundland, so these are littermates. The only difference between these littermates is that one is a knockout for TRP, so they're this light brown versus the dark black. So it's the same thing in poodles and in Shar-Peis. Furthermore, we can make a mouse knockout by PubMed, as I like to say. So in 1992, there was a mutation described in mice that is an arginine to cysteine mutation in the same domain of the protein, and it leads to what they call the brown, what I call a beautiful blonde mouse here, that is exactly the same mutation, but in mice. So it really kind of gives an example of how we can take this idea of beginning to focus on different human populations, some of the traits that we know may be subject to selection, and to really tease apart for the first time the genetic basis of some of the the coolest traits that segregate in our species. All right, so I want to summarize by first telling you that this idea of going out and doing whole genome mapping in domesticated plants and animals can work. It is quite economically feasible. So we've demonstrated this in dogs. We've demonstrated this now in rice, in grape, and we're really sort of just, beginning to open up the possibilities of genomic applications. Humans have dramatically shaped the dog genome, and I'm happy to tell folks more about it. We can actually clearly differentiate breed dogs from village dogs simply by looking at patterns of homozygosity. One of the things I didn't talk about is that the most critical part of all this is figuring out how to deal with the data. It's a huge amount of data, and there are real algorithmic challenges. So if anyone's interested in going into this area, I encourage you to learn about as much math and computer science as you can stomach, because it's really what's going to make a lot of this move forward, particularly as you start thinking about having 1,000 sequenced human genomes in your inbox. The other point I want to make is that model systems remain really key, right? So the only reason we were able to make sense of the human data is because we were working on dogs and because we were working on mice, but there are lots of other model systems that we really remain really important and critical to understand. So for those of you who are evolutionary biologists and you're working on some godforsaken frog species and you're thinking, oh my God, who's ever going to care? You never know. You can always stumble upon cool phenotypes, and if you begin to eke out the genetic basis, then they could turn out to be really important model systems. When we started working on dogs, everybody thought we were crazy. Who wants to work on the genetics of dogs? They're not a model organism. You can't make a dog knockout. They're not a good evolutionary biology system because they're selected. It's a totally dumb thing to do. We've had a bunch of cool science and nature papers, so we're kind of happy with the progress of that today. The other thing is that we're only now starting to really knock out some of the same traits in humans, and so we don't yet know. I mean, you would imagine, like, how do we not know the genetic basis of skin pigmentation variation in humans, of hair color, of eye color, of why pygmies are short and bantus are tall? How do we not know this, right? I mean, it just seems like these are the kinds of really basic, anthropologically interesting questions that we need to be able to understand. And the price of doing this is just dropping, right? We're really entering this $0 genome. So one of the things that to me has been really exciting is to really engage with anthropologists, people who work on the field, and think about where the intersection of genomics with anthropology can really move it forward. I really need to acknowledge a lot of the folks who were critical in all this work. Elaine Ostrander and Bob Wayne are my partners in all the CanMap Project. Even though I talked about it, it's really a three-way collaboration. It's just been a tremendous pleasure and honor to work with them and folks in their lab. There are a bunch of people in my lab who've been critical to all this work. Emer Penny and Sean Miles did all the collecting and analysis of the phenotype data for the humans. And then Adam and Lynn Lee did all the algorithmic work for dogs. And of course, I can't help but also thank all the people that have donated samples, and in particular the people of the Solomon Islands who were just so wonderfully gracious for hosting us and allowing us to work on this beautiful population. Thank you very much. Working. But I'll repeat your question.
So there was a proposal years ago here, people I know, to work on dogs breeds because their behavior is stereotyped. For example, that border collie won't go in the wire, the Portuguese water.
Yeah.
Can you use this approach?
So if you look carefully at our CanMap slide, we had something on prey drive behavior. Behavior has been particularly hard to tease apart, I must admit. We have worked on some stereotypical behavior that segregates within breeds. So we had some colleagues at Cornell who were working on what was known as Springer Spaniel rage. So this is a very characteristic, it's brains that happens in Springer Spaniels, look just like Cocker Spaniels. So you have a mental image of a Cocker Spaniel, you have a mental image of a Springer Spaniel. And these dogs have a very stereotypical owner-directed aggression that they'll, bark at the owner, but they'll still follow commands. So the dog is barking at the owner, but he'll sit, and it seems to be just something that's totally cross-wired.
Well, this is wonderful stuff.
And so we've been able to kind of get down, but it's a tough story because it traces back to a dog that won, what's it called, the Crufts Dog Show, or the big dog to Westminster. It won Westminster, and so everyone's like, hush, hush, 30% of Springer Spaniels are descended from that dog. But we're starting to get at it, and I think if we can crack behavioral genetics in any mammalian system, dogs will probably be the best.
Which dog is most closely related to the wolf?
So that's a great question. The question was, which dog breed is most closely related to the wolf? And the answer to that is that, in fact, it seems that all dog breeds are more closely related to each other than any of them is to the wolf. So they do form. So the Mastiffs are some of the most ancient dog breeds, but they're not the most closely related to the wolf. There are, you do see, for example, some parts of the genome where Huskies relate to wolf, but that's probably the regions of the genome that give them the phenotype that makes them look somewhat wolf-like. So it's hard to tell. The other thing that we see, and we've worked, for example, on black wolves. So it turns out that the mutation that gives rise to black wolves came from dogs. Okay, so there's also been a lot of integration between dogs and wolves, and different wolf populations today have a fair amount of dog in them. So you have to be a little bit careful when you're trying to do those sorts of analyses. Other questions? Yeah.
You mentioned the financial problems. I should think that various and sundry large medical people or groups would be interested in doing something like this, possibly testing raising resilience.
For dogs, you mean, or?
No, transferring this onto the medical field for.
Right. Yeah. So there's definitely there are big concerted efforts to go after some of the big heart disease, diabetes, and there's plenty of money going after that, and NIH has been great. And actually, so one of the nice things that's happened in human genetics over the last 10 years is that human genetics went from a field where everyone boarded their samples and said, no, these are my samples, and these are my samples, to say, you know what, the only way we're going to be able to make sense of this is by bringing all our samples together and building a consortia. So that's been really great, and NIH has basically really rewarded scientists who collaborated and brought samples together. The biggest problem is that most people have the samples from the people who are easiest to collect, right? And oftentimes, some of the populations that suffer the most in terms of health disparities are the ones that are most suspicious of participating. So African Americans, Hispanic Latinos, who have some of the worst rates of diabetes, for example, are reticent to participate for all kinds of really good historical reasons about what's happened in the past. So Native communities, for example, we really try to work closely with Native communities to try to engage them in medical genetic research and say, if you don't participate, you won't benefit, but we obviously also completely respect your desire not to participate. So here's some things that we could think about, here's some other things that and it's a really touchy issue, but a really important issue because, as I said, so it turns out I was going to talk a lot about human population genetics, and then I heard you guys talk about human population genetics, so I decided to talk about the dog stuff. But one of the things I'm most passionate about is trying to really enable a global human genomics and allow people to participate if they wish to participate. And so there are all kinds of interesting ethical issues to think about. How do you do that in a way where people have enough informed consent so that they can participate, where if you are going to be involved in a medical genetic study and they say they're going to put your genome online, well, is that a good thing? Probably not. I wouldn't put my genome online. There are all kinds of issues that arise in that, but it's a dialogue, and that's why it's really important to have geneticists involved, but not to leave it up to the geneticists, right? They're probably the last people you want to leave it up to. You need to have sociologists involved, ethicists involved. It's a tough problem. Yeah.
In
And in particular, so I would say methylation is one thing that's been put out there. Rare alleles are another. When people have looked at cognitive deficiencies, for example, one of the things that they've found now when they're able to look at other kinds of variation there is that often there are deletions. So there are often deletions that are segregating there that weren't well represented in the SNP polymorphisms or the DNA polymorphisms that were involved. So I think we're starting to piece apart both the contribution of rare and other kinds of structural variation. I think epigenetics is really fascinating potential, but I think you have to really have a kind of complete misreading of heritability in order for the epigenetics to sop up all the missing heritability. Right? For height, for example, like it's something like 80% is missing. Right? So it's hard to really figure out because the epigenetics are also polymorphisms that impact epigenetics. And so people have been looking at when you have these GWAS signals, they're in fact enriched for regions that impact the methylation markers. Right? So that's the genetics on the epigenetics, it's not epigenetics. So we can talk more about it, but I think you have to come up with it's an interesting hypothesis, but I'm not ready to bet the farm on that. Yeah?
What problems do you envisage with putting our own genome online?
What do I envision? No, so I think, I should say, okay, in disclosure, I have a project now where we're sequencing a bunch of different human genomes. And I think putting out anonymized human genomes is probably not a problem, right? You don't have associated polymorphisms with particular phenotypes, and in fact, we have to encourage people to participate in medical genetics research where the data will be either released without associated phenotypes or in what are known as data access controlled databases. So data access controlled databases are basically, you have to have some credentials in order to get access to the data. So you have to be somebody who's funded on an NIH grant, or you have to have sort of minimum clearance to get in. And I actually think that's a good idea, right? You don't want just anybody being able to use the data for whatever they want. In the same way, I don't put my anonymized credit card information out online, right? I just don't think looking forward, it's hard to predict and know what are the potential harms that could come of it. There are now really good laws that have been passed against genetic discrimination, but I think it's uncharted territory. So I think it's important that people understand what are all the issues involved. And we often don't know those.
Is medical insurance your main consideration at present? Give me some examples of what.
Well, so medical insurance is one of these dodgy areas where, in fact, it's not covered by current legislation. So that if you release information about your status, I'm not a lawyer. I'm not, in no way, shape, or form qualified to give legal advice. I'm just giving you one person's opinion. But we could consider, we could imagine situations where knowing your status for Alzheimer's or knowing your status for breast cancer, right, could lead to a situation where you would be denied medical coverage, right? And so it's this sort of fine line of have you been tested genetically for, and in fact, it'll happen in the form of a questionnaire, right? When you fill out a new insurance form, have you been tested for a particular disease? If you've had your whole genome sequenced, well, that's not really getting tested for that. But if you were sequenced because the doctor hypothesized, then is it a pre-existing condition? And what about a mutation you inherit? Is that a pre-existing condition if you don't have any signs of it at the time you enroll, right? And lawyers are sneaky, right? They're paid to be sneaky, right? And so there is actually not, you have to have the legal theory that underlies all this stuff is totally open. It's conflicting, and I think it's one of these things that will end up getting resolved in court, And
all the good
absolutely. Well, that's totally true. In fact, so the countries that have given the most breakthroughs have been Iceland and Finland and now England, right? Because they have really good, digital medical records, and in fact, they'll be the ones that are going to benefit the most by application of this technology into their populations. In fact, we've started working with Mexico because Mexico is a much saner country to think about application and a lot of these issues than the U.S. because it's a very well organized, centralized, medical system as opposed to the U.S. where it's a mess. Yeah. That's a great question. It turns out this is a phenotype that changes with time, so the hair gets darker as they get older. And we had IRB approval to study the change, obviously. Yeah.
Excuse, could you backtrack just a moment to give a brief definition of epigenetics for us amateurs?
Sure.
Related to the previous discussion of behavioral genetics.
Okay, so epigenetics is like pornography, right? Everyone's got their own definition. I'll give you a couple of examples of what people mean by epigenetics. I would say that the most common definition is it is something that's not just inherited by the DNA, but it often can go across multiple generations. So there are what are called methylation marks on the genome that suppress the expression of a gene, and that often gets transmitted from generation to generation, and it can change over time, right? Other epigenetic examples are you might inherit, there are genes that you only express the maternal versus the paternal copy, right? That's kind of an imprinting effect, and some, that can also impact things. So it's basically anything that gets inherited that isn't just plainly in the DNA, and that can change over a short course of generations. I'm sure there are people in this room who know much more about epigenetics, so if someone wants to chime in with a better definition. Yeah?
I've always been curious. You've really presented an excellent synopsis here. Incredibly varied type of dogs that there are. But if one thinks about the number of domesticated cats in the world,
Yes.
Is humans simply not messed with domesticated cats over the last 15,000 years, or why are there not?
Yeah, so that's a good question. I mean, it's interesting, right? There are different theories that have been put forward. First, cats are just kind of ornery, right? They've domesticated humans. Humans haven't domesticated cats. They certainly don't seem to show the kind of body size variation that dogs show. And one hypothesis is that, and I didn't go into a lot of the molecular mechanisms, we think that many of these dog mutations actually have to do with really active transposable elements that are jumping into genes and knocking out the effects. So they may have different kinds of mutations that have big effects. That was a particularly kind of crazy hypothesis initially. No, maybe not so crazy. If you look at, horses, right? Horses also, they vary in size, but you don't have achondrodysplastic horses, right? You don't have horses with foreshortened faces, right? But there are cats with foreshortened faces, right? So some, it's a different set of questions. But I would bet that the mutations, once you start looking at cats with foreshortened faces, they may have mutations in the same gene or same pathway as dogs with foreshortened faces.
But cats don't demonstrate the same growth differential that the dogs clearly do.
That's true. But cats vary. There's some big cats and there's some little cats. They're just not, super tiny cats. Yeah. I'm a dog person. What can I say? All right. Well, thank you very much. It's been a pleasure.